Hey gang, hope you're enjoying your election day off. I want to post this other video for when I go out of town Thursday and Friday. You'll have some stuff to work with. So some of the stuff on here is going to be a repeat of the notes of the organelles you've got and of the drawings and the four basic areas that you got in class on Monday. So some of this is a repeat, so you can just ignore those parts. Here are the learning targets for section 1, just two basic ones. So pause it and write those down. Here's a diagram that shows lots of things, but it goes over those four basic areas again that you took down, that you put down on the diagram yesterday. Here are those terms and a written definition of those, so please write those down. These are not on your list of organelles. So this is about the nucleus. Remember the parts of the nucleus we drew yesterday? The nucleus has multiple parts. It has the nuclear envelope, or the membrane, which is a double layer. It has holes in that envelope which allow some things to pass out, but not everything. We'll learn more about that later. It has a nucleolus somewhere in the middle or close to the middle. It's a very dark staining body. Its function is to produce ribosomes, and it also has DNA, usually in the form of chromatin, which is long, stringy, thin strands of DNA. But every once in a while, right before it's getting ready to go through cell division, it will turn into a thicker form of DNA called chromosomes. We'll study chromosomes when we get to the mitosis unit. The cell membrane is another area of the cell. It's that very thin, flexible, fatty layer outside the cell. And remember, all cells have cell membranes. Cell wall. Cell walls are found in two types of eukaryotic cells, plants and fungus, and then in bacteria or prokaryotic cells. It's a thick protective layer. And then we have all those organelles that were on that one chart that I gave you. Remember, it's very important for you to understand the structure and their function. Here's two learning targets that kind of lends itself to that, so be sure you get those down. This kind of tells you what an organelle is. I'm not going to read it to you. It gives you the analogy of organs in our body versus organelles in a cell. This tells you that there's some of them that have a membrane surrounding them and some don't. This, by the way, is a key feature or a key difference in prokaryotics and eukaryotic cells, which we'll see just in a minute. So here's what I really want to talk about that I haven't talked about in class. When we look at the rough endoplasmic reticulum, and remember it's rough because it has ribosomes attached to it, under a microscope it appears bumpy or rough. Then you have this Golgi apparatus, which consists of these membranous sacs stacked on top of each other. The Golgi apparatus tends to operate like a factory. The rough ER makes a protein. Here you can see it as this greenish structure. It's transported by pinching off a piece of the ER's membrane, the protein inside it. That little piece of membrane is called a vesicle. It transports it to the Golgi apparatus, where it's passed from Golgi body to Golgi body, and then eventually repackaged into another piece of membrane, this time from the Golgi body. Basically, just the end of this body pinches off, and the contents are contained inside. Remember, the green thing is representing the protein. And then that vesicle can either move to the cell membrane and export that protein, or it can be used within that cell. So together, the ER, the Golgi apparatus works together to transport proteins that are made in the cell to another location. So when we talk about comparing cell types, there's two ways of doing that: plant versus animal cells, and prokaryotic cells versus eukaryotic cells. You and I are eukaryotes. We are an organism that have eukaryotic cells. So let's take a look at the comparisons. First, we'll do plant and animal cells. Okay, there's the two ways to compare. Here's plant versus animal. This is pretty simple. I'm not going to read this to you. Just know those differences. Okay. More importantly, are the words prokaryote and eukaryote. So here's a breakdown of how these words translate from the Greek into English. So it turns out that karyo means nut or kernel, which sometimes kind of relates to things that are in the center of something. And the person who came up with this word to stand for this kind of cell took nut or kernel and roughly translated it to nucleus, because nucleus is kind of like in the center of a cell. Pro in this case doesn't mean professional, doesn't mean getting paid for. It means before. It's like a timeline thing. Pro means before. So when you put those two words together, it turns out that before the nucleus means no nucleus. So these were the first cells on Earth. These are very simple cells, and they do not have a nucleus. Eukaryotic cells, and I didn't put a picture of this because that's what you drew the other day. U means true, of course. Karyon means nucleus again. So these cells have a true nucleus. And they also have lots of parts. Let's go on to finish the comparison. Here's a classic Venn diagram, and you can see the differences on either side of the Venn diagram with the similarities in the middle. But I want you to look over here at eukaryotes. They're complex. They have membrane-bound organelles. Some of them have cell walls. Over here, no membrane-bound organelles. These are very simple cells, and they're very small in size. This is only found in one kingdom of life. This is found in the other 4 kingdoms of life. Okay? That's it. Be sure you take good notes. I will be checking notebooks before the 9 weeks is over. Have a good day.